know what they're doing. So, but thank you. Michaela Gunther is going to be introducing our speakers tonight. She is a professor in the wildlife department at Cal Poly Humboldt, and her research focuses on the behavioral ecology of mammals. She's been on our Conservation Council since it started in 2011 and helps organize this lecture series every year. And so we're very grateful to all the hard work that she puts into the committee. Thank you so much.
Thanks,
all right,
so goodness, right. Thank you for that kind introduction. It's introducing the introducer. So that's really nice. That's the kettle. Okay. I'm so happy to be here this evening, and the weather isn't too bad. We had a lot of rain last month, so that was nice. Thanks all for coming out. I love being part of this community, of this zoo committee, and of this organization for getting this community conservation lecture series going. It's been a really fun part of my contribution working here with the zoo staff and getting community members. They're so hungry for this information, so excited. We get such a great turnout every month. And so I really enjoy my contributions to helping organize this lecture series for the zoo. So tonight I'm especially happy to introduce two of my colleagues. So as Ruth mentioned, I'm a faculty member in the Department of Wildlife at Cal Poly Humboldt. Many of you affectionately may remember it as Humboldt State University. I've invited two of my colleagues from the University College of Natural Resources and Sciences to talk today about some of the research that they're doing in our community. So it's a team lecture so that they can cover a lot of information. Don't worry, we'll get out of here on time. We used to have a clock here.
No, I know. Now it's over there though.
Oh, okay. Okay. So Dr. José Marín Jarrín is originally from Ecuador, did his graduate work here in the US, actually close by in Oregon. Worked in Ecuador for the Charles Darwin Foundation, did a lot of work on coast seamounts and seabirds and sharks, and working out in the Galapagos among other coastal areas. Has been here at Cal Poly Humboldt working interested in local fisheries, especially high use fisheries, and has been involved in these marine protected areas that have been established here on the coast. And we were really interested to hear about the successes of these marine protected areas. And I approached Jose to come and please talk about this fishery side. And we talked about how nice it would be to have even a bigger story about the marine protected areas. And we invited Dr. Mark Colwell, who is a professor emeritus in the Department of Wildlife, which means he just retired this past year after 35 years of teaching in all sorts of ornithology-related courses in our department. And he has been working his whole research life on shorebirds all over the West Coast, let's say all over the place, but really here focusing on the endangered snowy plover population that we have here, and also recently with the marine protected areas. So they're going to give a team talk covering some of the fish and some of the bird stories in the marine protected areas. And without further ado, I'm going to turn it over to these two. Thank you so much for joining us.
Yep, right behind you.
There.
Okay.
So, good evening. Thank you so much for inviting us. My name is Jose, and like Dr. Micaela mentioned, I'm going to be giving the fish side of the work, and then Mark's going to come in second and talk about the fishes. So I just wanted to briefly begin with introducing my lab. This is the Marine Fisheries Ecology Lab. We have a really nice, diverse group of graduate students, two of which are here, and then a few other grad students as well. And we also have a technician, Katie, who was my first grad student here at Humboldt, and who's a little bit of her data I'm going to present. And then we also have a lab manager and research associate who's also here, Michelle. So I'm going to introduce several topics. You may already be familiar with them, but just to make sure that we're all on the same page, we're going to be talking a lot about MPAs, and a common definition is they're discrete geographic marine areas designed to protect or conserve marine life and habitat. You may be familiar with this map. It comes from CDF&W's website, right, indicating the different MPAs that we have along the coast. And their definition, they're slightly different in the take and what you can do in each one of them. Now, we have had about 60 MPAs in California for a while, but in 1999, the state passed a Marine Life Protection Act, which required us to double the number of MPAs on our coast. So we went from 60-something to 123 or so. The process was complex. I was not here for it, but I understand there's even a documentary if you're interested in it. There were a lot of stakeholders and governments involved. But basically, they got to us last. So the deadline was 2012, and they got ours established in December of 2012, so just before the deadline. And very little work has been done, especially on the water side. In fact, because of this, once they were defined, the first work on the fish side occurred in 2014 and 2015, describing the biodiversity of our waters. So our MPAs are fairly new and quite poorly explored. Okay, so why MPAs? Why do so many governments try to declare them and to form them? Well, because most research suggests that when you form MPAs, they can allow fish to grow larger, that there be more fish and be more diverse fish communities. So this is some data that's often shown. This is a paper that was published a few decades ago now. And what you're seeing, this is a literature review, which means that the scientists looked through all the scientific literature and found all the papers that were working on MPAs, and particularly in fish, and then looked at if MPAs had fish with higher biomass, higher density, so number of fish, their size, and the diversity of the fish communities. And on average, what they found was that the biomass of the fish increased almost 5 times, the density increased almost 2 times, the size increased almost 30%, and the diversity increased 20%. What was also very interesting is that you might notice a few of the points are also on the negative side, and some are right at the 0 line, suggesting that in some cases, the number of fish, their size, and everything else actually decreased or stayed the same, so there was no change. There's been a lot of work since then, and what they have found is that in order to ensure the effectiveness of MPAs, there are certain characteristics that need to be met. In general, they have to be big, old, no-take, enforced, and isolated. Those first few ones are the ones that we are easiest to take care of, as you can imagine. So they have to be big enough so that your fish doesn't leave the MPA, right, and gets caught by the fishery or something like that. Otherwise, it's really hard to protect them if they leave. Okay. So the second big topic I want to introduce is a habitat that's near and dear to my heart. These are sandy beaches and their surf zones. This habitat makes up about 50% of the world's open coastline. And in the Pacific Northwest, which we are part of, right, it's over 60%, over 70% in certain parts of the coast. This is an environment, the surf zone, that is open to the water, so often it's constantly rechanging with waves, but is semi-enclosed to organisms because of their habitat, because of their behavior. And the surf zone itself extends from the shoreline to the outermost breaker. And if you've ever stood on Clam Beach, I'm sure many of you have, if not all of you, right? You've noticed there's multiple places where the wave breaks. So the farthest out, that's where the surf zone ends. Why do we care about surf zones? They're incredibly complex, sandy beaches and their surf zones. So they provide a habitat for inverts, fish, birds, and other vertebrates, including us. They get their energy from 2 main sources. The first one is from coastal ocean processes, which on our coast is mostly upwelling. And that's when really deep water comes to the surface. And that brings really cold water that's also very rich in nutrients. Those nutrients are really important for phytoplankton that then goes crazy and reproduces like mad, means a lot of food for sand crabs, so mole crabs, which ends up being food for surf perch and shorebirds. And of course, we enjoy the surf perch ourselves. The other source of nutrients is from macrophyte racks. So when you walk the beach, all those dead plants, right? They're technically not plants, right? Most of them are photosynthetic organisms. The phycologists get really upset when we call them plants, right? They're not plants. But all of those photosynthetic organisms that end up on the beach that are really good food for amphipods and shorebirds, actually amphipods also for surf perch. But those racks, that rack's not coming from the beach. It's coming from other habitats. So estuaries, wetlands, rocky intertidal, and kelp forests, right? That's where they grow. They get torn off by big waves and they end up on our beaches. And that's how they're accessed by the amphipods. So it's a fairly complex system that's connected to other systems as well. And besides that, they provide ecosystem services that we can monetize, like nutrient cycling, wildlife support, water filtration, and even nursery habitat for inverts and fish. So really complex and important systems. Okay, so just a bunch of pretty pictures about different surf zones around the world. They can have fairly high biomasses of fish, as you can see in this picture, which attracts fishers, both commercial and recreational. We mostly think of it as a habitat for small fish, but the more we look, the more we see really big fish. So mostly chondrichthyes, so your sharks, rays, skates, that come into the surf zone to feed on the inverts and on the other bony fishes. Of course, fishes are not the only ones attracted. We all are to it, to the coast, right? And that has led to a very large population of humans, right, living on the coast, which of course requires lots of resources, human development in general, right, to make space for all of us, but also the need for recreation and food and all these other things that we need. So, and I apologize, I'm really nerdy when it comes to sandy beaches. So I'm gonna take you to how we categorize sandy beaches. I've been doing this for over 10 years now. I actually started with sandy beaches as in my undergrad. So it's been a while, I'm 44, so it's been a while looking at sandy beaches. But there are many ways to classify sandy beaches. This is way more than you need to know about them, but there's many ways. One is dependent on how energy is released from waves. And it can go from reflective to dissipative beaches. And there's a bunch of intermediate types in the middle. But reflective are beaches where the energy of the wave is reflected off the slope or the shoreline. So it's a very steep slope, right? That's what you see here. And the wave gets really close to shore and literally bounces off the slope and bounces away. Usually there's only one breaker, and because of the energy that gets all the way to the coast, the sand is fairly coarse because the finer sand can't settle. And so it's constantly being moved offshore. Like I mentioned, several intermediate states. And then at the other side are what are called dissipative beaches. So that's your Clam Beach, your Mad River Beach. Those are dissipative beaches. They're fairly long. There's multiple breakers, as you can see in this diagram, right? And so the energy is dissipated every time it breaks. So the wave comes in, breaks once, releases some energy, breaks another time, and breaks another time. The sand tends to be finer, right? Because the energy is dissipated, and so it allows for that sediment to accrete here, leading us to have finer sediment. Now, beaches can change over seasons. So in the wintertime with the big waves, right? You'll notice Clam Beach, Mad River Beach are much flatter. That's because the bigger waves drag all that sediment off and make them flat. But during the summer, waves are smaller, so it allows for the sediment to come back, and that's what makes them a little more steeper, right, in the summertime. So again, more than you needed to know about sandy beaches, but now you know. Now you might be wondering, okay, so 60% of our coast is sandy beach surf zone, so why should we care, right? There's so much of it. Well, because they're under threat throughout the world. There's a fair amount of work that's been done on this. So Omar Lefebvre from Uruguay and his colleagues published this paper a little over 15 years ago. And what they looked at was the different threats, and they plotted them in space and in time. And so what they found is that there are many things that can impact sandy beach surf zones, going from small impacts like recreation and ORVs, right? They have a small impact in space and in time, right? If you're driving in some kind of vehicle on the beach, right? You're limited to how far you can drive. And the impact is short-lived. But there are other things that have a much bigger impact. So pollution, nourishment and grooming, beach mining and exploitation, for example. There are things like coastal engineering and urban development, right? That have a fairly small space impact, but will be present for decades, if not centuries. And then what's looming on all of us, right, and throughout the world is climate change and sea level rise. Every sandy beach in the world is threatened because of sea level rise. If you think of most of our beaches, there's mountains behind them. So the sandy beach can't move backwards because there's a mountain that prevents them from moving. It's one of the big problems for many habitats that are on the coast is that they can't migrate inland, especially on our coast. Another way of thinking about it is what's happened in the past. So now I'm showing you backwards in time. So this is year from the 1200s to 2000, and this is the intensity of the coastal development. And so in the 1200s and 1300s, 1400s, what we were doing to the sandy beaches had a very small impact. So we had small-scale dune stabilization projects, early protection structures. But as our technology improved and our populations increased, our impact grew larger. We had large-scale dune stabilization, major shore protection measures like jetties and beach fills and marinas and ORVs. Humboldt Bay was not an open estuary year-round, right? It was probably a lagoon that we now keep open because of the jetties. How does that impact how sediment moves? It's a big part of the equation, right? And then more recently, we have international tourism. We love visitors, right? It's a big part of our economy, but that means more resources. It means the potential for invasive species, right? Riding on their shoes, et cetera, et cetera. So the impact has increased through time since, and now for several centuries. Okay. So how does Mark and I come into? So in the MLPA, when the act was declared, one of the things that it required is for the state to monitor the MPAs. And so that's where we came in, right? There's funding for monitoring MPAs, all of them in all different habitats, and there's multiple projects, many of them going on at Humboldt, in fact. And so for the fish side, the objectives were to describe the fish in macro-inverted community and sandy beach surf zones, and to study if there's a spatial or temporal variation. In particular, the state was interested in knowing if the MPAs were benefiting these communities. So, just looking a little washed, but on the fish side, we have 8 different sites, and we go from almost the California border all the way down to just north of the mouth of the San Francisco Bay. We have what we refer to as pairs of beaches, so one MPA and what we call a reference site. So these are beaches that are very similar physically to the MPA. That allows us to compare them and see if the MPAs are truly benefiting the fish that are inhabiting them. So all the beaches that you see with an asterisk, that's what they're highlighting. Those are the MPAs. So just north of here, we have Reading Rock, which is the MPA, and Gold Bluffs, which is its reference site. Mad River, just north of here, right, and Samoa Beach, those are the next pair of beaches, Samoa being the MPA. In Mendocino County, we have 10 Mile Beach with the MPA and Virgin Creek, the reference site. And in Doran and Drake's Beach in Sonoma County and in Marin County, or as I like to call it, my county, right? It's my last name. So we sample all these beaches mostly during the summer, about 3 times a year, depending on funding. And the cool thing is that we get to hire a bunch of undergrads who then we send off to sample these beaches. They get really good experience and a little bit of money in their pockets. We use a plethora of methods to study these fish, and a lot of this is because it's really hard to sample surf zones, it's dangerous, and fish are really fast and can get away from you really easily. So one method that we use is referred to as BRUVs, or baited remote underwater video. And that's just a really fancy way of calling GoPros, which is what we have here. You can see them here attached to weights. This is just your dumbbell that you might use to lift weights, right? It has a PVC pipe and a bait bag here. And so we deploy these in the surf zone at about 1.5 meters, which is like 6 feet to about 8 feet. We leave them for an hour, and the GoPro just records whatever fish comes to try to munch on our bait. It has a little buoy on the top, and you can see them here, which allows us to know where they were so we can go pick them up after. Now, this sounds awesome, and it is, but I'm just gonna show you just a short snippet of what the video looks like. If you've ever been to our surf zones, you've noticed it's really turbid, and we have hundreds of hours of these, right? That's why I get grad students, so I don't have to look at it myself. but it should play. I mean, what keeps us going is that every now and again, you see really cool things. Come on. So anyway, the point is very turbid, and then you see something like that. This is actually a river otter. This is Mad River Beach. I don't know if you've walked Mad River, they're often there. And so there it goes again. And so very difficult, right? You need specialized people who have certain technical expertise, because the fish all look the same, right? Especially when it's that turbid. And so it's really hard and it takes a fair amount of time. We're actually experimenting, this kind of pains me, but we're experimenting with AI to see if we can improve that. It pains me because being a field person, right? It's hard to then give it up to something else. It's not even human. But anyways, a topic for another day. But that's one method. The second is more traditional. It's a beach seine. This is just a big net that we drag into the water, we deploy, and then we pull it back on shore. And of course, I have a video for you, and I found this, and I thought Mark would get a kick out of this. Actually, Mark took this video of us many years ago. We were just starting. And so you can see us pulling the net back on shore. It's a fairly short beach short seine for those of you who have used beach seine. And that's because in the water, it functions as a sail. It literally just drags you. And so if it's a big seine, it drags you offshore, and it's really dangerous. So instead, we use this small beach seine. And I don't know if you saw the previous photo, but the sides have holes, and that allows us to keep the seine on the ground so none of the fish get out underneath, because the fish are quite smart and are good at avoiding capture. The final method is another traditional method. This is hook and line, so angling. So we send people out there to fish, mostly for surf perch. We use a Carolina rig, which is fairly standard for surf perch fishing. This work only happens in Humboldt, so a lot of the data that you'll see will be from all the beaches, but the hook and line work is just from Humboldt sites. Okay, last thing I'm gonna tell you on methods. This is statistics, and I know it's kind of late for talking about math, but so historically, right, we use what are called univariate statistics. That means there was just one response variable. So for example, if I wanted to compare this side of the audience to that side of the audience, I could measure everyone's height, right? And compare the two and see which one was higher. But what happens if I am also interested in the length of your hair, the length of your arms and your legs? All of a sudden I have multiple response variables. Now, because of the computing power it requires, in the past, we would just first compare height, then compare hair length, then compare arm length, and so on. The problem with carrying out multiple statistical tests is that there's something called false positives. So every time we run a statistical test, there's the potential that we run into one of these false positives. If we run many statistical tests, the chance that we run into a false positive increases. So the fewer the number of tests, the better, basically. So that's the issue that we have. And so we use, among the multivariate statistics, one that's called within the ordinations, and it's in particular, we use non-metric multidimensional scaling. And you don't need to know anything more than that for now, but that's what we use. So the way that this works is picture 3 lakes, A, B, and C. A has circles and stars, B has circles, stars, and squares, and C has triangles and squares. So if you look at what fish each one has, you will notice that A and B are the most similar, right? They share 2 species, circles and stars. So if you think of them, because they're more similar in terms of distance, those 2 points should be closer together. The next one, if you notice, B and C share 1 species. So not as much as A and B, but still share 1 species. So their location, their distance should be short, but not as short as the previous 2 comparisons. And then finally, A and C share no species, right? So they should be the farthest apart from each other. So if we think of these samples or lakes in terms of distances and not in terms of species and abundances, we can plot them in an XY form. So in 2 dimensions, instead of these many dimensions that would require having multiple variables. So then we can take the samples out of the equation altogether and just think of them as distances in space. And that's what we see here. And that allows you then, say, if we had 4 samples, we could even compare them. So we can visualize them and statistically compare the different groups. And this is the kind of work that you can do with multivariate statistics. The problem is, in the past, we did this all by hand, right? And it was a lot of work. Now R does it in 3 or 4 lines of code. So it's way easier now. Okay. So finally, the results. What did we do? So here's some pretty pictures that we have taken over time. We've caught over 24 species of inverts, including Dungeness crab. These are actually the most abundant group in our sampling, which is great because that's the largest fishery on the coast these days, right? Lots of bony fish, in particular surf perch, lots of chondrichthyes, bat rays, and leopard sharks, and even a dogfish. And we also see marine mammals, so river otters and harbor seals are often out there. So a fair diversity of species. So how do we plot this? So this is that same NMDS that I mentioned. Each of these points is a sample. And here are 8 beaches. And so what Katie did was she plotted them all on one figure. And what we were hoping was all the data from the MPA would fall on one side, all the data from the reference sites would fall on the other. So showing that there was a difference between MPAs and reference sites. That is not what we saw. So for example, Drake's and Doran, so if you remember that Sonoma and Marin County beaches, right, are actually right on top of each other. One's the MPA, the other's the reference site. And we saw this with every beach that we saw, and they were basically one on top of the other, showing that there was no difference between MPAs and reference sites. The cool about these type of statistics is that it can also tell you what species were driving these patterns. And so what we found was that at Virgin Creek, we caught a lot of kelp crabs. At Drake's Beach, we had an unidentified crab and red rock crab. And at Doran, we had bat rays and hermit crabs in the sandy beach. Probably lost hermit crabs. They shouldn't be there, right? It's not a good site for them. So this is the RUV data, right? All the video. When we looked at the SANE data, we saw a similar pattern. Again, Drake's and Doran separate from each other, but very close to each other, and everyone else just piled on top of each other. We found no difference between the MPAs and the reference sites. But again, we could see what was driving the differences, and barred surf perch and calico surf perch and Drake's, half smelt in Doran, and staghorn sculpin at 10 Mile. If you don't know what staghorn sculpin, if you've ever walked on sandy beaches and you see little fishes taking off, those are probably staghorn sculpin. They're one of my favorite fish. If you catch them, it's a male, it'll hum at you because it thinks that you're another male trying to mate with it. And so it's telling you, hey, no, that's not working here. And so it's asking you to let it go. So those are staghorn sculpins. They're really fun. They're one of my favorite fish. Really tough little buggers. Okay, so Katie was like, no evidence. So what do we do? So she decided to look at the diet. So she looked at stomach content of these fish, and that's what you're seeing here. Again, notice they're all piled on top of each other. There was no difference between MPAs and reference sites. She even separated red tails, and this is just red tails, between sizes. So she looked at small red tails, medium red tails, and large red tails. Again, very little difference. It turns out red tails, just like mole crabs, they're just a one-trick pony kind of fish. They really like mole crabs, and so they eat big mole crabs or little mole crabs, and that's it. And so no difference. She was hoping that MPAs could have more mole crabs than reference sites. At least from the diet analysis, it doesn't seem to be the case. So that's Katie's thesis, and she did way more than that, but I'm just showing you the cliff notes, so to speak. Then Noah came, and he thought, well, maybe it's because we're looking at the whole fish community. What if we look at just one fish species, right? No more multivariates, just univariate NL. So that's what he did. He looked at red tail and silver surf perch. He just offended last week. He's still sleeping probably, right? Because we made him work a lot this last few months.
He's on Zoom.
What's that?
On Zoom.
Oh, he's on Zoom? Oh. In that case, hey, don't forget to finish those comments, okay? We're not done. Just say. Okay, so what he did was he threw more complicated statistics at them, and we don't need to go into those. I don't understand them, so I can't explain. So, but I trust that that our statisticians explain correctly to know. But what he looked at was count, length of the fish, weight of the fish, and their ages. And then he looked to see if the MPAs had higher numbers than the reference sites. And so what he, for count, for example, he found that he looked at fish if they were immature, female or male, and he found that there were statistically more males than females on our sandy beach surf zones. This is good because these fish are viviparous. That means they give live birth. They literally give birth like we do. And so if we're catching more males, that's better for the population because we're letting the females stay and keep giving birth, which is really important for a fishery. He also looked at years. He looked at 3 years, 22, 21, and 20, and he found that there was a statistical difference. 22 had higher counts than any of the other 2 years. And then finally, he looked at by site, right? So here are the 4 sites of Humboldt, Samoa, and Mad River, the Pear, right, and Reading Rock and Gold Bluff. And he did find a difference, but he found that the MPA in the north, the most north MPA, the Reading Rock MPA, had more fish than Samoa. As we go through the data, you'll notice that this pattern keeps showing up. The beaches that are farthest away from the Tri-Cities, Eureka, Arcata, and McKinleyville, have higher numbers than the ones that are closer to us. This is actually fairly common in these kind of studies, right? If we don't have a lot of time, we're not gonna drive an hour to Gold Bluffs to fish. We're gonna drive 15 minutes to Mad River to fish instead. So we tend to, right, reduce the population that are closest to our bigger towns. Assuming you're not like me and you can't catch anything, in which case they're safe from you, right? So he looked at length as well, and these are called violin plots. And so the way to think about these plots is the places where the figure is widest, it means we have more data. The places where it's finest, it means we have less data. So this is a way to show you how much data he was working with. And so here he's separating by site, right, 4 times, and then immature fish, female fish, and male fish. And again, we're looking at length. On average, the fish that we were catching, 25 centimeters, so about a foot long. And he found some evidence of MPA benefits. So he found that there were longer immature fish in Reading Rock than in Gold Bluffs. So that's the pair that we were interested in. But everything else pointed to no benefit. So he found longer female fish at Gold Bluffs, and longer male fish at Reading Rock than at Mad River. Again, the 2 distant beaches had longer fish than the close beaches. Again, by weight, right? Again, the 4 sites on the bottom, immature, female, and male. He found that the fish at Reading Rock, the females were heavier than the Mad River males, and so were Gold Bluffs males. So again, Reading Rock, Gold Bluffs, heavier than the Mad River. The farther beaches, heavier fish than the close beaches. And then finally, he looked at age, and he found that the fishes could be between 0 and 7 years old. We think red tails can live to be 14 years old. The average of the fish that we caught were about 3 years. They can become reproductive at 2. So for the most part, we're catching reproductive fish. But again, he found the same pattern. And I'm just going to scroll through because this is probably going to get repetitive. But the point is, the fish that are in these Reading Rock and Gold Bluff, the far beaches tend to be older than the ones that are in closer beaches. So the pattern seems to be that it's the distance from the towns that is really affecting the populations. So to summarize, sandy beaches are awesome, right? And this is why we spend a lot of time studying these habitats. But this, since 2019, this has been the first comprehensive study of fish in Northern California. We so far have found little evidence of MPA benefit. It could be because the red tails are moving too much. There's some work that shows that they can move multiple kilometers in just a few days. So the MPAs might not be big enough to be protecting them. The other thing we are finding is that there's more education and outreach necessary. So thank you to the zoo for conducting this kind of projects. We several times found people fishing in MPAs. What was interesting is the moment you told them they were MPAs, they immediately realized what they were doing and they would just move and fish somewhere else. So we don't think it was people trying to poach. It was just people not knowing enough. And so if you've noticed, if you walk by MPAs, there's no signage on the parking lot. 10 Mile has it, but nowhere else has it. So some kind of signage, I think, would help a lot. The northern sites seem to have longer, heavier, and older red-tailed surf perch. It could be the distance from town, but it also, there were some differences in the steepness of the slope and in the sediment. The two northern beaches were steeper and had thicker grain size. We think that that means that because it's steeper, bigger fish can get closer to shore, and that allows the fishers to access them. So it might be not so much a biology thing, but an availability to the fishery kind of thing. We also found a few other things with the environmental conditions, but I won't bore you about that, at least not this time. So I'll stop there and I'll ask Mark to take over.
All right, well, this is coming up. I'd like to thank Jose for the nice introduction and also acknowledge lots of students and community members who played a role in the work that I'm going to talk about, especially a grad student who is my last student, Jay Blair, and you'll see some of his data from the surf zone that Jose emphasized, but I'm going to move to the terrestrial aspect of this. And I suppose Jose and I could have a couple of arguments already about the structure of the trophic community that exists here. Maybe in the wash zone that he's sanding for fish in, and it is more complex. But when I think of the sandy beach habitat that you see here, which is on that stretch of beach from Petrolia, you know, that last stretch right before you go to Petrolia, this particular image was photographed a few years back in the context of snowy plover work, exemplifies the autochthonous source of the detritus from offshore kelp beds that ends up on the beach and contributes to a relatively simple food ladder. I think not so much web-like, but ladder-like in that the detritivores that feed on this are those beach amphipods. I'll show you a photo or two of them in a little bit, but then on them feed the shorebirds that I've studied for 40-something years. And then peregrine falcons, for instance, are at the top of that food ladder. So I'm going to talk about brown algae as the foundation, and it is sourced from rocky intertidal habitat offshore. And I'm going to avoid MPAs entirely in this presentation and just talk about sandy beaches. And I'll do so with two species or groups, snowy plovers, which I worked on for 20 years with students, and talk about the relationship between brown algae and a curious statistical result that we found that you'll see momentarily in their reproductive success. And then I'll turn to the more recent work that Jay Blair and I have done with regard to the brown algae, the invertebrates, and the shorebirds that feed on those invertebrates. So there's a distributional map for snowy plovers, and you know where we are. So on the Pacific coast, and the purple is that narrow or blue, depending on your perspective. Breeding range for that resident population here in Humboldt County. It also breeds as far north as Washington and down to Baja. That's a threatened species, and again, I worked on it for 20 years. So this is a little busy. I'm going to show you two images of Humboldt County beaches, sandy ocean-fronting beaches. So here's where the big rack line of that brown algae was down in McNutt Gulch. And then from Centerville north to, point your finger.
Yes,
Yeah.
should be the top one.
Sorry. Yeah, up to Clam Beach. And then this inset is the lagoons and Gold Bluff Beach. So there are a whole bunch of little circles there. Some of them are filled black, others are white. Every one of the black circles had at least one pair of breeding snowy plovers on that beach in the 20 years. And you can see that there are a lot of black circles showing the presence of snowy plovers, but there are a lot of empty circles showing in 20 years we never had plovers breeding there. So what's interesting is the plovers are on these beaches breeding, and they're there year round. They're feeding off the detritivores that feed on the brown algae that accumulates on the beach, right? It's Still haven't shown you the photo yet, but it's coming up. And they especially, plovers feed on them at night when the amphipods are quite busy. If you go out there during the day and kick over some of the sea palms, Postelsi or something, you know, there'll be a bunch of them that'll team and bounce around. If you go out there early in the morning, right after dawn, they're just still bouncing around. They're nocturnal and they start burrowing, presumably because of predation by shorebirds. But the brown algae accumulates on the beach, and this is simply a presence, absence image, and I'm gonna take all of the open circles out and relate the food to plover breeding success in this image. Sorry. There we go. All right, so all the empty circles are gone, but you've still got circles that are either white, gray, or black. The white circles show where plovers bred and had zero reproductive success, did not fledge chicks, and there's a bunch of them, right? The gray show the locations at which plovers had 0 to 1 chick on average. And the black ones, filled circles, are those where plovers did exceptionally well, better than what you would want for the recovery of this threatened species. So when we mined through the data, we used to collect a lot of data just walking down the beach on the abundance of brown algae. One of the strongest relationships was that where you had more brown algae, you fledged more plover chicks. So whatever's washing up on the beach is providing food, presumably, for these small plovers. That's not to say there aren't other things that affect, but it's food, and those amphipods are a big part of the food consumed by big plovers, adults, and the chicks. In fact, sometimes the amphipods are so big that the birds reject them. There we go. That's an amphipod bundle, and I'm not an amphipod expert, but you know, sometimes you see this and I'm thinking it's a mating swarm. But they also accumulate like this on top of fresh brown algae on the beach. So that's more than a dinner for a plover, big or little, that's a lot of food, right? So in terms of the trophic structure of the food ladder on the beach, the brown algae serves as the base for it. These are the detritomores, the next level up. Then come the shorebirds that feed on them, and peregrine falcons that feed on the shorebird. All right, there's a top-down picture emphasizing, there's a plover leg here with a band on it, and all the feathers are plucked. This is a juvenile bird that fledged in Oregon, showed up on Clam Beach and got eaten by a peregrine falcon out there. And it just emphasizes the top level of the food ladder. So if we're talking about the beaches and not the wash zone, it's sort of the strand of the detritus that washes up on the beach, including that brown algae-based food ladder. What Jade Blair did as part of his research was to survey beaches up and down Humboldt County coast and record, I think there's 20,000 bird observations in his data set. And you can see that the majority of them certainly the vast majority, 60%, are shorebirds. The other species that are on that list, gulls in particular, often are just loafing on the beach. They're not feeding. The shorebirds are actively feeding oftentimes. There are times when they're roosting, but most of these birds, the shorebirds, which include lots of sandpipers, are picking at debris and eating those amphipods. There's some other other invertebrates out there, but this is predominantly a bird community dominated by shorebirds. All right, Jose used the same figure with spatial and temporal axes here. In this case, small to large, spatial extent, and predictable and long to ephemeral and short. These ellipses, I'll come back to at the end to emphasize this, but they represent what algae do in this system, right? They're patchy, and when it gets deposited on the beach, it stays there, right? Fairly limited in extent. Amphipods map onto them in space and time because that's the amphipod's principal food. The shorebirds eat amphipods, but they get pushed around by that top ellipse. And peregrines are here year round. This is one of the few areas in the world where peregrines, there's a population of resident breeding peregrines. And at this time of year, there are even more peregrines because they're migrants that come in here, but they're moving around in search of their prey, shorebirds. I'll come back to this. Okay, so a few simple graphics here. We've got algae at the base and the detritivores, amphipods in particular. And so these samples here show a very strong relationship along the beaches. This is a log scale, so that's 100 times more than that. But the point being is strong correlation. Where you have algae, no surprise, lots of amphipods, right? Pretty clear correlation. I'm not sure how many would be under those big bundles that were the size of a Volkswagen Bug. But it could be off the charts. So lots of variation in amphipod abundance, but strongly correlated with the detritivores' food. All right, and so the next tier up is the amphipods feeding shorebirds, a variety of sandpipers in particular. There is a significant relationship here, but it is not like the last one, right? It's all over the place. Amphipods on the x-axis, shorebirds, all of them grouped on the y. And that is significant, but there's a lot of slop to that. I have a question mark here because we don't have the data on the rarest species in the food ladder to actually evaluate the relationship between shorebirds and the predators being falcons. And it's mostly peregrine falcons. They showed up so infrequently in our work that we couldn't analyze this. But still, this relationship exists to some extent, and the birds, shorebirds, move around in response to their pursuit by peregrines in particular. And that's not a local bird, but I mentioned before, there's a resident population. My colleague Jeff Black had a student working on the peregrines locally, and these birds are resident year-round on their territories, although during the non-breeding season, they could be anywhere along the coast. That's rare worldwide, it only occurs here. And the point being is these birds move a lot in a long distance in pursuit of their prey. So I think that's the explanation for the absence of a relationship. We just don't have data to assess it. Okay, so back to this, just to finish. Algae wash up on the beach, pretty restricted, and they don't really move much, right? Other than a little bit of wave action. It doesn't really move from one day to the next. Amphipods, really clear as detritivores feeding on the algae. The shorebirds are present, certainly, and correlate to some extent with their principal prey, amphipods, but they're moving around a lot. And they move around a lot, partly because of the predators, but also because many of these sandpipers are out on the beach at high tide, feeding in the rack line. And why are they there at high tide? Because Humboldt Bay is inundated at a high tide, and their principal feeding habitat is inundated. They can't access the mudflats on the bay. Lots of them come onto the beaches to feed due west of the bay. I don't have the data here in the slide, but there is clearly a relationship between diversity and abundance of shorebirds due west of Humboldt Bay compared to what's north along the beaches, as far north as the Klamath and south to Mattole. Along those beaches, you don't have the birds there to the density and diversity that you do due west of the bay. So complex interactions going on between this simple ladder, trophic ladder. So I'll just finish with that photo of, again, the stupendous amount of brown algae that accumulates on the beach. occasionally. This is my Gulch again, and then principal detritivores that are feeding them. I think I have time for some, and Jose does too. Yep.
Study following the big blob in 2015 and 16, and I wonder, and they showed devastation for the whole ecosystem. I wonder if you saw that here, and does it linger?
You're talking about the warm water blob
Yeah.
in the Gulf of Alaska,
Right.
and this stupendous result for king crabs, for instance, and some other seabirds showing I don't know. That's sort of a larger spatial scale than the local scale I'm talking about. So I'm not sure how it would relate to, the localized beaches that I'm talking.
You didn't see decreases in birds during that time?
Not that I could attribute to that.
Okay.
We found a correlation, but with smelt. Smelt have a fairly short life. The night smelt lived 3 years, surf smelt lived 5 years. And we noticed a decrease in the size of night smelt in particular after the blob, because it was 2 years of blob and 1 El Nino as well. And so we think that because night smelt have 3 years and there were 3 bad years, it completely tanked their populations. And so they're just coming back now.
But they are coming back.
Well, at least for now.
Kind of.
Yeah, with 3 years, it's hard.
Surf
And, night smelt.
Next.
Surf smelt seem to be actually, Daniel, who's sitting in the back, is looking at surf smelt right now. And so it was a okay year for cactus, but we don't have anything to compare it to. That's the other problem, is that we have no data to compare it with. So it's hard to say if it's good or bad. Thank you. Quick question.
Question for you, Jose. You were surprised, maybe disappointed, weren't yet apparent differences between the MPAs and their reference sites. And you mentioned maybe the fishery being. But is it also possible, so it's a two-part question, do you also compare this to the other MPA paired sites? I know the species are gonna be different and so forth, but I'm wondering if things are good up here, maybe there isn't a lot of fishing pressure, and maybe that it's great to have MPAs across the California coast, but up here, the reference sites are good. So it's hard to say if there's no difference because they're not protecting anything and they're not working, or if we don't need them as much. I don't even know if you could even distinguish that, but is there like a population pressure? Can you look at a correlation between the population fishing pressure?
That's a really good question. So we are part of, Mark and I are part of a larger group of collaborators. And so we have another group in Central California, another one in Southern California. And the folks in Southern California have seen benefits of MPAs. And they do attribute it to the fact that there's just a lot more people there and there are a lot more anglers there, right? Their weather also allows them to angle all year round, ours doesn't. So there is the hypothesis that it could be fishing pressure. Unfortunately, we don't have enough data on fishing pressure up here. They do in Central California, and they have found a relationship between how well the MPAs do and the amount of fishers. And so it's unclear if that's the case. One of the things, actually, Mark asked this question during Noah's defense is, could it be that the MPAs are benefiting fish, but they're just being eaten because there's a lot of top predators around? And that's something we have not explored. It gets tricky because to study that, you have to like tether fish and then record it when they get eaten. It's kind of gruesome too, right? And so you can imagine the permits for those of us who have to get permits. It would be quite a bear. But there are several avenues that we haven't explored that could show that they are providing benefit. The other thing is that the red tails seem to be one of the most mobile surf perch of all. Some have been clocked between here and like Southern Oregon. And silvers, they can go out to 100 meters in depth. So there is not, it wouldn't stop them. And I didn't mention this in my presentation, but our MPAs are about 2 by 3 kilometers. So like 2 miles by 2 miles. Yeah, they get around that pretty easily. It works really well for rockfish, for example, right? Rockfish, they tag them and like, you catch them here and there, you come back a year later and they're still in the same rock. So it works really well for rockfish, but surffish might not be the right species to test this. It's really cool.
Explain to that. Has anyone done a study on the size of an MPA and the effectiveness of that size? Because like you said, I mean, if it was a terrestrial ecosystem and you took like a 2 by 2 mile reserve, you're not gonna really see much protection there, right? Like, all of our really big animal parks are, you know, hundreds of miles above.
Yeah, so it was interesting. So I wasn't here when all the discussion went on how big and how far apart, but the thought was that by having a network of MPAs instead of one really big one, it could allow for dispersal. So dispersal is when the larvae or the babies basically move by the currents, and so where they end up, that's the dispersal. And so if you place them at a certain distance from each other, the babies from one could go to the other MPA and so on. And so the idea was that the network would be more beneficial than just one big MPA. There's some evidence that a network of MPAs is also more resilient in the face of climate change, right? Because if you have one every so kilometers as waters warm south, the fish could move, just leapfrog from MPA to MPA kind of a thing. It's of course way more complex than that and blah, 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 but that was the thought process in that. And they threw everything we had at it, oceanography, models and everything, and that's how they came up with the size and the distance from each other. But yeah, it's a really good point.
Yeah, I have a question from I've been part of a volunteer project where we walk the sandy beach once a month.
Mhm.
And what I've been kind of surprised at when we talk about brown kelp is that I guess I'm not surprised that it's brown kelp, but it seems like often there's an influx of seagrass. It seems like it's transient, but I'm kind of surprised that the seagrass itself didn't have an effect.
Yeah, I know exactly what you're talking about. When the eelgrass from the bay washes out of the bay, it's typically May or June. But there's also an egregia that's a intertidal, rocky intertidal seagrass. Those do not grow amphipods. They just, if you go down the beach and kick over the eelgrass, nothing. As opposed to the amphipods are specifically feeding on brown algae.
Yeah.
A variety of types.
Yeah, it's just so interesting, there's so much of it, but nothing
Yeah.
seems to be making.
It's really episodic, but it's May, June, and I'm not sure what goes on in the bay, but it just belches eelgrass on the beaches.
We do have a couple online questions, if you don't mind, we go over a couple minutes.
Sure.
Okay. Christy's wondering if any research has shown what an ideal size would be for MPAs that would benefit these species you're studying.
There has been work, and unfortunately, it all depends on the objective of the MPA, whether it's to protect one species or all species. And so our MPAs were more for all species, and that's when it gets really tricky because it's not going to be one size fits all. And so that's why they went with a network as well. That was the other argument for the network. So unfortunately, there's no good answer.
And then Christy also had a sympathetic question, wondered if you used motion detection cameras or software, or whether your poor grad students had to watch all those videos all the way through.
So we are exploring AI right now, but no, unfortunately, my students had to watch all of them. Yeah. So they went through a lot of podcasts and a lot of Netflix. Yeah. It can't be too exciting that it makes you stop paying attention of the video, but it can't be too boring that you get bored of what the video. So it's tricky.
All right. Well, thank you so much for coming tonight. We
Thank you.
all appreciate it.